Hello, everyone. Welcome. Thank you for joining us today. Welcome to our first online session of the Molecular Connectivity Online Series, or MCOS. My name is Sildes from New Jersey Institute of Technology. I'm also the chair of the Outreach Council of the Molecular Connectivity Working Group. Our group includes researchers from diverse backgrounds, such as biomedicine, health sciences, neuroscience, and physics. We aim to advance molecular imaging as a tool to study brain connectivity. We organize Molecular Connectivity Online Series as a platform to exchange ideas and to promote scientific research on molecular connectivity. We plan to host this online series monthly, every third Friday of the month. You can go to our website, subscribe to our newsletters, and follow us on Twitter and LinkedIn to get the latest information about this online series. Today, I'm thrilled to introduce our first speaker, Dr. Arianna Sala. Dr. Sala is a FNRS postdoctoral research fellow at the Coma Science Group, University of Lake, Belgium. With a background in molecular medicine and a focus on molecular biomarkers in Alzheimer's diseases, she has brought her expertise to study the brain glucose metabolism and metabolic networks in disorders of consciousness. Dr. Sala is also a co-founder of the Molecular Connectivity Working Group. In today's talk, Dr. Sala will share insights on brain connectomics, time for a molecular imaging perspective. Before we begin, a quick disclaimer. The talk will be about 30 minutes, followed by ample time for discussions. We will record the talk for further reference. You can ask questions during the talk by using the chat function, or you can raise your hand and ask directly during the discussion session. Once again, thank you for being with us, and welcome Dr. Arianna Sala. Thank you.
Thanks a lot. So let's see if I can share my screen once again. Okay. You can see correctly? Okay, great. So thanks a lot for being here. It's a lot of people, so well, I hope I will do a good job in introducing this topic. So most of the talk will be really based on what we described in this position paper that came out well, almost 1 year ago in Trends in Cognitive Sciences. So anything I might say, you will find it more more extensively here. So we'll talk about connectivity. I guess most of you are really familiar with what connectivity is. I must say, let's say, in this talk, I will use as reference the definition that was given by Carl Friston, considering connectivity as statistical dependencies between neurophysiological events happening in different brain regions. So behind connectivity, there is this idea of an interaction between different regions, and sometimes also of let's say, capturing the communication between different brain regions. Today, of course, we talk about molecular connectivity, so connectivity coupled with PET, well, molecular imaging, which can be both PET or SPECT imaging. For today, I will really focus on positron emission tomography. Okay, so what about using PET to study brain connectivity? So the history of brain connectivity actually starts with EEG, when in the 50s we had the first studies about cross-correlation. So we don't speak about connectivity yet at the time, but about cross-correlation, with the same idea of capturing the interaction between neural activity of different brain regions. In 1984, we have the first studies of PET interregional correlation, which again is not called connectivity because the concept simply doesn't exist yet, but it still is based on the idea of capturing the interactions between different brain regions. Then we have the first fMRI connectivity studies that are actually called indeed functional connectivity. Then structural MRI, DTI for structural connectivity, MEG, and NIRS studies. And in the history of brain connectivity, we have a few milestones that we could define as milestones. That is, first of all, the formal definition of functional connectivity, which actually, as you can see here in green, was based on PET imaging. So connectivity was actually first defined based on water PET to capture perfusion. Then we have the definition in the end of the 90s of the default mode network, which is again based on meta-analysis of mainly perfusion PET, water PET data. Then the connectome was defined, the resting state networks were defined based on functional MRI, and the connectome was defined based on structural MRI. So two, let's say, of the most important milestones in the history of brain connectivity are actually based on PET data. So where are we now? So what happened to PET in the last year? So currently, the majority of brain connectivity studies are actually based on functional MRI that make up around 2/3 of all the literature about connectivity. And instead, brain connectivity studies based on PET make up about, let's say, 3–4% of the literature. So they're really, let's say, a minority. In this talk, I would like to outline why it still makes sense to use PET to study brain connectivity, what we can actually do with PET, because we have different tracers targeting different biological targets. So we really have very different types of brain connectivity based on PET. And then I will discuss how we can do it. So coming to the why. I think a very important point I would like to make is that with PET, at least with some tracers, we can really go to, let's say, a bit more directly on studying the neural substrates we are interested about. And this is why, I think there is this idea that, let's say, neural communication is something that is mainly electrical, but actually it's not, or let's say it's not only, because we have, in the postsynaptic neuron, we have the electrical signaling that elicits some, release of neurochemical packets of information into the synaptic terminals that gets picked up by the presynaptic terminals and transformed again into electrical signaling. So we have really this double nature of neural communication that is both electrical and biochemical. And with PET, we can target many aspects of the biochemical component of neural communication. So, we have very different tracers, and I will come back to these a bit more in details later, but just to give an overview for those of you that are not familiar with PET, we can measure many different things. Some targets are at the level of the blood, like, for example, we can measure blood perfusion. Other tracers like FDG-PET that measures glucose metabolism targets events happening in astrocytes and neurons. And then we have many different tracers to actually investigate directly postsynaptic and presynaptic neurotransmission. So we have a measure that is more direct, for example, compared to BOLD as measured by fMRI, which again is the predominant tool that we use to measure brain connectivity. And a very important point is that it is here there is I mess up a bit with the slides, but the thing is that PET is highly reproducible. So if you go to FDG-PET, for example, we have 90%, the intraclass correlation coefficient is of 0.9, which is compared to BOLD fMRI, and in particular ALFF is much higher. It's been reported to be 0.53. Also, there is a good point to discuss is the spatial resolution of PET, which is, well, not so bad. With the current scanners, we speak about 4.3 millimeters. And we have, for example, here you have a comparison between different neurophysiological tools. So here you have a simulation of, some signals in this hypothetical 3D cube. Here you see what you would see using structural MRI. Here with diffusion MRI, and from here on with more functional techniques, for example, fMRI, then PET, MEG, NIRS, and EEG. So as you can see, the resolution, well, this is how it would look like. This is the gold standard, and now it would look like based on PET. And what does it mean? Is it a good resolution or not? Well, it corresponds. So with PET, we are measuring for the average of around 2 million neurons. For comparison with fMRI, every voxel contains averages of signals from around 800,000 neurons. So with any neurophysiological techniques that we have, we are several orders of magnitude away from this a single unit measurement. At the time, from a point of view of temporal resolution, we are in the order of seconds to minutes with PET. Here you see a representation on a logarithmic scale where the reference is the resolution of fMRI, which is around 2.5 seconds. As you can see, we are pretty far away from the, let's say, temporal resolution of neural communication if we think of it as an action potential, which is around 1 millisecond. With fMRI, we are capturing averages of around 2,500 action potentials, simplifying a lot, of course. With PET, it depends. So we have different protocols of acquisitions. In some cases, we use a static PET, which has a resolution of some minutes. But we have also some functional PET protocols that allow to derive frames with a duration of 16 seconds. So again, we have very different time scales across different techniques, and I guess also different spatial scales. So does it make sense to go, what is the temporal spatial resolution that makes sense to investigate, to use? I would say here, no neurophysiological technique that we have available right now is, can do everything. I think we look at things at very different spatial temporal scales with very different biological targets, and each of these neurophysiological techniques allow us to see one little part of the elephant. And this is very important because, I mean, with just one technique, we cannot just capture the complexity of neural communication and brain function more in general. So to obtain an accurate model of brain function, we need to gather information for all different techniques, including molecular information. You can see here some representations of connectomes that you might obtain. This is actually real connectomes. We obtain the data from different collaborators. You can see how different the connectomes are when using different structural, functional, and molecular techniques. So we can get unique information from all the different techniques that we have available. For what? So as I mentioned, there is different types of brain connectivity that we can investigate based on PET. So we have, here you see what targets we can investigate based on all non-molecular neurophysiological techniques. For example, NIRS, fMRI, MEG, EEG, and structural and diffusion MRI. So we are really targeting very different things. For example, with MRI, we're measuring a signal in the blood vessels. With EEG and MEG, we are going at the level of the neurons, of the postsynaptic neurons. With PET, we have many different tracers. Some tracer, again, can measure targets in the blood, like perfusion and oxygen metabolism. Other tracer measure instead glucose metabolism, which is a bit more direct index of neural activity compared to Ball signal, as it captures the glucose metabolism that is happening at the level of astrocytes and neurons, and is directly coupled to neural communication, and in particular excitatory communication and glutamatergic signaling. When we investigate brain connectivity based on these neural activity markers, we are basically referring at a brain connectivity similar to how it was defined in the 90s by Friston based on perfusion PET data, where essentially here we investigate interactions between brain regions based on the relationship between our markers, our molecular markers, like for example perfusion or metabolism, and electrical activity. Like, for example, in this case, it was referring to cross-spectral densities as based by other neurophysiological markers. So in this sense, this type of molecular connectivity is rather similar to the standard, as a traditional functional connectivity. And there were very old studies from the 80s that investigate, let's say, the connectome, even if it's not how it was called at the time, that show, for example, strongest correlations or connections between homologous regions in the two hemispheres, and in particular between the frontal and parietal regions, which is not dissimilar to what has been replicated later on with different techniques. We also have neurotransmission markers that we can investigate, and we can go at a postsynaptic level to investigate the different types of receptors that are expressed at the postsynaptic levels and at the presynaptic level, which is something quite peculiar of PET, because the other techniques capture events that are mainly reflective of postsynaptic activity, but with PET we can also go at the presynaptic level, and we can investigate activity of transporters, neurotransmission synthesis, and that are indeed reflective of presynaptic activity. So we are really going into the biochemical point side of neural communication. And like, what kind of connectivity is this? Like, does it make sense to speak about connectivity with this kind of markers? And I would like to explain a bit using an example that was initially provided by Andreas Hahn. You can still see the full example explained in a symposium that we had at OHBM a couple of years ago. So the idea here is that the expression, for example, of some transporters will be between one region and the other in the brain will be coupled. How is that? So let's consider a serotoninergic system at the level of the raphe nuclei that projects towards the full brain at subcortical and cortical level. So a very important player in the serotoninergic transmission is the serotoninergic transporter, which is part of the presynaptic machinery of serotonin. So in the raphe nuclei, we have a high expression of serotoninergic transporter, which essentially takes the serotonin from the synaptic cleft back to the presynaptic neuron. When there is a high expression of this transporter, we will have a decrease of serotonin in the synaptic cleft, which will lead in a, let's say, internal regulation mechanism to a decrease in these 1A receptors, which are autoceptors that are, let's say, get downregulated when there is a low expression of serotonin. So when we have this kind of situation, we will have an increased firing in the raphe nucleus, which will result in a higher availability of serotonin at the projection, so far away, for example, let's say, cortical level. This will be coupled to also an increase in the activity of the serotoninergic transporter and a decrease in the expression of the 1A receptor that are indeed a regulator based on the availability of the serotonin and the projection site. So essentially, this is all to say that some events that are very far away might actually be coupled, where increased expression of the serotoninergic transporter in the raphe nuclei will result in increased expression of the serotoninergic transporter also at the level of the projections. And indeed, it was shown that there is a correlation or connectivity between the expression of the serotoninergic transporter in the dorsal raphe nuclei and at subcortical level in the ventral striatum as a projection. And this is present in healthy controls, but this is lost in conditions like, for example, major depression. So again, so we can really interpret this as dysregulation of the serotoninergic system and altered connectivity or interaction between the nucleus and its projection. We also have another type of connectivity, which I'm not sure if we should call it connectivity or not, actually. But let's say it's this is related to instead all the targets that are related to proteinopathies. So with PET, we can also measure pathological processes in, for example, tau tangles accumulations of amyloid plaques, which are the hallmarks of Alzheimer's disease. And there has been indeed quite a few studies using these markers, these biological targets, to investigate brain networks. And why does it make sense to do it? Well, if we consider tau pathology, for example, we have that it has been shown that tau, these pathological aggregates of tau tend to propagate from one neuron to the other through synaptic connections. So if two neurons are functionally connected, so if there is essentially a synapsis between one and the other, one misfolded tau protein might be transmitted from one neuron to the next. So what happens is that essentially from a starting seed where there is this misfolded tau accumulation, we can have a propagation through connected brain regions. And this has been shown both at the level of anatomical projections and also of functional projections. So here again, this is not connectivity in the sense of communication, but in the sense that there is an interaction between different brain regions that is mediated by neural activity and anatomical connections in determining the spread of pathological proteins. And we can use network approaches to predict how these proteins will spread in the brain. We are back to the how. So last point. So how can we, well, investigate brain connectivity? So we have different methods. They're not really dissimilar to what is normally used with fMRI. If any of you is familiar with those, we have, for example, seed-based networks where we can investigate the pattern of connections from a specific region to the rest of the brain. We have whole brain approaches like independent or principal component analysis, where we decompose the signal in the whole brain, so at a voxel level, to identify different networks or components that are, let's say, made of voxels whose signals tend to be, well, let's say, similar. Let's say the variation of the signal is similar within voxels that belong to the same network, the same component, and not really to the rest of the brain. And then we have approaches that are region-based, like, for example, ROI to ROI correlations that allow us to investigate either the whole brain or specific circuits. So it's not very different, but there is something that is actually pretty much different. And this is because with PET data, we can have very different types of PET data. Some data are, let's say, depending on the mode of acquisition, we can obtain data that, let's say, that we can use for estimation of connectivity at an intra-subject level. So what we have is that, well, we have our subject that undergoes a PET scan, and we are able, let's say, and we want to, let's say, investigate connectivity between the thalamus and the occipital cortex. So we do our acquisition protocol, which will be a dynamic protocol or let's say functional PET protocol, and we measure the uptake in these two regions across time. So in the same participant, we have essentially derive a time series for each of the regions that we are interested in. So this is essentially, we investigate brain connectivity as statistical dependencies between time series, looking at, let's say, similarity and consistencies in the variation of the signal across time in the same subject. But most commonly, what we actually have with PET imaging, or at least this is what has been used so far, is inter-subject modeling. What it means is that we have different subjects that are relatively similar between each other. So let's say we have all healthy controls between 20 and 40 years old. And again, we want to investigate connectivity between these two regions, but in this case, we have a static PET protocol that gives us essentially only one image per subject. So we will measure the uptake in these two regions for subject one, the second subject, third, fourth, and so on. And what we have here, it's not a time series, but a subject series where, again, we want to look at consistencies in the variation of the signal, but not across time, but across subjects. So this is the inter-subject approach, which is commonly used to assess brain connectivity when using PET imaging that most often, in 85% of cases, relies on static images, so one measurement per subject. So I guess Many of you wonder, like, is it the same to use these inter-subject or intra-subject approaches? Since the 90s, we've been more often used to see brain connectivity as studies based on time series, so intra-subject correlations. So there has been a few studies, not nearly enough, but a few studies trying to investigate this. And there was one study by Sharna Jamadar, using structure, comparing the connectivity matrices that we can get based on a static PET protocol. So this is inter-subject modeling against a functional PET protocol. So intra-subject modeling with PET and functional MRI. In this case, the results were that functional PET and functional MRI results were more similar than static PET and functional PET results. So suggesting that there is only a limited degree of periodicity between inter-subject and intra-subject results. At the same time, I guess it's a bit hard to derive this kind of conclusions from these results because of the different time scales in the different measurements. I mean, we have, for functional PET, we are using a temporal resolution of 16 seconds, while for static PETs, we're using a temporal resolution of several minutes. So it will be, let's say, it's hard to tell whether the results, this mismatch is due indeed to the different approach used or simply a different time scale that is used. And there was quite interesting results that were presented by Cindy a couple of years ago. And you have here actually the QR code to the full presentation that he gave. But essentially what he did is that he took the Artin dataset that contains a lot of longitudinal scans from, different populations. In this case, it was 72 healthy controls that have undergone between 5 and 9 scans available. So we have, of course, different subjects, meaning that we can estimate connectivity inter-subject with a cross-individual approach, so inter-subject level. And we also have, for the same participants, 5 to 9 scans available for a within individual assessment. The duration of the scans, it's the same in this case, and it showed that when we estimate connectivity based on inter-subject and inter-subject modeling, we have actually a pretty good correspondence between these two, with a correlation of 0.77 between the two measurements, suggesting that indeed there is, if we remove the component of different temporal scales, there is some similarities because between what we can get at an inter-subject level and at an intra-subject level. So, and this is still, I guess, an open issue, and there is a lot actually that is left to do in the field. One important thing that I think we should do, and actually we are doing, is agreeing on a nomenclature. As I mentioned today, I spoke during the presentation about connectivity, but different terms have been used in the past. And today, still some people prefer to speak about covariance, some connectivity, so it's very, let's say, varied. And here we have, and I don't want to spoil anything, there will be a big announcement in the coming days, but part of our group is actually working on organizing a consensus meeting to agree on this important aspect. Super important point, validation. Related also to what I just showed about inter-subject correspondence between these approaches, but very important point is validation. That actually has many different facets to tackle. What is validation? For sure, we want to use approaches that are robust, that are precise, and are reproducible. There is some work based on this. For example, Mattia Veronese showed that there is a good reproducibility with some tracer and with some approaches, at least. For PET connectivity, for example, you see here at F-DOPA how is the distribution of correlation between test and retest. Validation is not only technical, it also means proving that what we are measuring actually matches the gold standard. So, for example, are we really measuring something reflective of neural communication? In this case, it's a bit very hard to do because, like, what is the gold standard with brain connectivity? We don't have a gold standard measure that tell us, this is neural communication in its purest form. We have fMRI, we have EEG, but these are all different proxies of connectivity, different parts of the elephant, right? There was a study by Igor Yapyshev that actually proved that, now assuming that functional connectivity should have a structural substrate, It proved that functional connectivity based on fMRI, you can see here in blue, and molecular connectivity based on FDG PET, here in orange, have a similar underlying structural substrate. So the connection that we see are not really random, but are based on something anatomical, structural, that could serve to some extent as a gold standard. There is a lot of things to do. A very important point, as I said, there is different approaches, different methods, and different tracers to measure connectivity. So I say it is still missing a systematic review and a gap analysis of what has been done in terms of validation for all types of validation, for all tracer and all methods. And this is, again, our team is working on that. We're also designing validation studies to fill the gaps in the literature. And here we have a lot of other team members working on it too, with the idea that all of these will be needed to finally reach best methodological practices and indications of what should we use to get the answers that we want. And again, the final aim is indeed to achieve, well, cross-modal integration to get to a better representation of, a more accurate representation of models of brain function. So combining, well, first of all, understanding in which relationship are all the markers that we have. So which parts of the elephant is each marker actually showing to us? What is the relationship between all of these? And then develop methods to increase the robustness, to better understand the biochemical substrates of the connections we are studying, and get also to describe brain function in terms of causality. All things that we can do with better technical instruments, but also with a better understanding, I think, of what is every technique that we have available bringing to the table. So lots of things to do. I would actually thank all the people that were involved in this conceptual paper and are working all together now to try and tackle these, well, complex questions. Also our other members that joined more recently. I also would like to thank, that I didn't mention yet, Mary, Matej, and Michael. You didn't saw in the presentation, but they're part of our outreach group. And we've seen that, well, you know very well at this point, they've been working to put up together this molecular connectivity series to start discussing about these important questions. And thank you very much for listening.